And we're live. Welcome everyone to the Evidence Reasons Academy. I'm joined here by Katie, and we're just going to do the show and start sort of where we left off on nuclear localization signals. And like a lot of research, we may be just visiting all sorts of places on the internet and in the realm of ideas to try to come up with something, and we may or may not find things, and just like a treasure hunt, there might be a lot of days we don't find a thing, and that could be this session. But it might be informative to the audience what the process of this sort of research is like. And so where we left off, how about I let Katie summarize what she thought our last session was about and some of the work she did in the interim, and then I'll give my take on it.
Okay. Well, last session we were looking at the BLAST database. It's a database, right? Yeah. And we were looking at different protein. Well, we were looking at a protein sequence from a certain fungi, which I'm blanking on what it's called again.
Aspergillus.
Aspergillus,
yes.
We were looking at was that the topoisomerase sequence?
Yes. Exactly, topoisomerase 2. Very good.
And so then we were looking at where the localization signal was on that protein sequence. But really it was just to get comfortable looking at or just getting around the BLAST database. And we also used the UniProt database to look at other organisms, I believe.
Yes, excellent summary. And when I looked at the recording of our video last time, I was mentioning the basic amino acids, and I said lysine and arginine. He said the one with H, and I said, I don't know. I have to credit you. You're spot on. It was H, histidine. So there are 3 basic amino acids, lysine, arginine, and histidine. And my jaw dropped. I said, Katie remembered that. And I forgot. And I was the one that was showing it to you. So I wanted to tell you how impressed I was.
Thanks.
So as much as I'd like to, we might do a little, if we start drawing blanks in the first half hour, I just might take us to just do some basic biochemistry and organic chemistry, just to take a break, just so we could feel like we accomplished something. Okay. Mm-hmm. So maybe we'll be lucky. So why don't you give me a report of what you found? So we agreed that we were going to try to look for databases that on nuclear localization signals. So why don't you give a little report on what you found?
Yeah, so when I did a little bit of research into databases, I found 2 different databases. They both they started with an acronym. I can look it up really quick. But neither of them are actually active anymore. One of them was through the NIH. And the other one, I'm not sure what that one was through. But the point is that neither of the databases work. When you try to click on a link towards them, they'll send you to like a warning kind of signal that the website doesn't exist anymore, or there was an error in the search.
Actually, when you feel comfortable sharing your screen, if you know what I mean, if you could share it when you're giving the names of these, just so I could have it also on recording, that I could see, compare it to any of my searches.
Yeah.
So in the meantime, I'll talk to the audience. So just take it easy as you bring that up.
Okay.
I'd mentioned to Katie backstage that even though these are very important to the cell, they're not immediately important, ironically, to the medical community. And like, how can something so important not be important to the medical community? And my speculation on this is if something is broken in the nuclear localization, transport, and export, um, the thing will be so dead it's not even born. And hence, there's nothing modern medicine could do anyway for that. And hence, I think, there's just not been a lot of interest. Excuse me, that's a spammer. Let me get rid of it. Then let me turn off my phone. I forgot to turn off my phone. Okay, I behaved. I wanted to curse and swear whenever I get a spam call. But okay, I just shut off my phone. So so for that reason, there may not be a lot of investment, except they may want to study more about, the major reason someone would be interested in this is just to, study the architecture of the cell, which always counts for something. But where a lot of the money and grants go is like, does your project look like it's going to go toward creating a new medical treatment or cure? So that's just sort of the way it goes. I mean, we have to be grateful that money has been invested in research that didn't have any immediate obvious benefit. Probably the most famous example was Michael Faraday, like say 170 years ago or more, or around that much. He was researching electricity, and a woman asked him, what good is electricity? And he said, Madam, what good is a baby? And so because of Michael Faraday, we have everything in the modern world that relates to electricity. So that's kind of like, this question of nuclear localization, will it have any immediate benefit? And I could say I doubt it, except it would be of interest to the intelligent design, the creationist community, and the evolutionary biology community, what we're doing. So kind of the benefit is, we're basically now pioneers in the field since these databases are dead.
Mm-hmm.
And we'll be collecting these localization signals and kind of making our own collection. So have you been able, do you have a screen
Yeah,
share?
I
Okay.
can share the screen.
All right, great, thanks.
Okay. Okay, can you see my screen?
It's about to come up. Just one second. StreamYard is a little slow.
Okay.
I'll add NLSDB, database of nuclear. Look at that.
So this is the first one. This is just kind of a page, but you see that it has like the URL right here. And if I were to try to click on the URL, it says the page can't be found.
So it was a project.
Yeah, this one on the web, like
Oh
on
my
the browser,
goodness.
it said it's through University of Pittsburgh.
Oh, can you scroll down?
Mm-hmm.
Last update, 4/21/2014.
Yeah.
That's 11 years ago.
Yeah.
Actually, no, I'm really grateful you found this. You have such a nice voice. I really mean it. I cringe when I hear my voice on the internet. Could you read the highlights for our audience?
Yeah, I can read it. So NLSDB is a database of nuclear localization signals and of nuclear proteins targeted to the nucleus by NLS motifs. The database contains 114 experimentally determined NLS nuclear localization signals that were obtained through an extensive literature search using, I don't know what that is, times 91 in, what is that short for?
Okay. This is called X91 means how many, when we do a sequencing analysis, first for DNA, and then you can translate the DNA, you can predict it into what amino acids it may code for.
Mm-hmm.
So when they say times 91, that means um, how many experiments you've run to confirm that you really have a sequence. Now, when it says in silico, that means on computer. And
Okay.
so that's kind of funny because um, you're basically making a computer simulation, not a physical experiment.
Mm-hmm.
But then they extended it to um, ex- experimental results. So they I guess they tried to mutate this on the computer, and we
Mm-hmm.
would have to actually talk to the investigators or read their literature what that really means. So that's not unusual. I will tell you, sometimes they're such in a rush to describe what they did, they won't necessarily define their terms. They just want to get it out there, and
Mm-hmm.
then if someone's really interested, they can contact the researchers or look up. So it's not. That's a long answer to say it's not entirely clear, but times 91 usually means multiple passes of something. So 91 passes. So go ahead.
Okay. This set was extended to 308 experimental and potential nuclear localization signals. This final set matched over 43% of all known nuclear proteins and matches no currently known non-nuclear protein. The nuclear localization database contains over 6,000 predicted nuclear proteins, and they're targeting signals from the PDB and Swiss Tremble databases.
Okay,
Maybe.
so I'll stop you there.
Yeah.
PDB is the protein data bank, which has the X-ray crystallography. Swiss Prot is now Uniprot.
Oh, okay.
So now, okay, now this is good. See, now you're getting familiar with kind of the terminology. So when we were looking at Uniprot last week, do
Mm-hmm.
you or the week before? Remember how excited I was when I said you could actually see the protein structure on your system with the ribbons and everything?
Mm-hmm.
That's generated by Protein Data Bank, the PDB.
Oh, okay.
See, this is good because you can actually use the knowledge you've already learned in just 3 weeks to start reading stuff like this. I just had to do a little bit of translation.
Mm-hmm.
So now you can actually read this and understand most of what it's saying. So go ahead.
Okay. The database also contains over 12,500 predicted nuclear proteins from 6 entirely sequenced eukaryotic proteomes. I think I'm saying that right.
Proteomes.
Proteomes. Okay. Homo sapiens, Musmuselkis, Drosophila melanogaster. Oh, I don't know this other one. I'm
Okay,
not going to
so
try to pronounce the rest of them.
I think I don't know what musculus is. I'm embarrassed. You know Drosophila, right?
I do know Drosophila, yeah.
So what is it? You tell the audience.
It's fruit flies.
Well done. Okay, C. elegans. I can't even pronounce that first word.
That one sounds familiar. I feel like I
That's a
might.
worm.
Oh, it's a
That's a
worm.
worm.
Okay.
I don't know if it's a nematode, but don't you love all these Latin sounding names that they just
Yeah.
make? Um, and then Arabidopsis thaliana. Can you guess what that is?
I don't know.
It's a plant.
Oh, okay.
Are you into plants?
I
Uh,
don't have a lot of experience with them, no.
and then Saccharomyces cerevisiae. I think that's yeast.
Okay. That would make sense.
I'll go ahead and read the last, and I'm going to turn on the AC a little bit, so sorry for the noise.
Okay. Nuclear localization signals motifs often co-localize with DNA binding regions. This observation was also used to annotate over 1,500 DNA binding proteins.
Okay, so let me try to give my take on what it says. I'm not saying I'm 100% right, but I hope I'm close.
Okay.
Um, So remember, even when you do blast sequences for proteins, it's not necessarily that they actually have definitely that it's the protein. Remember that they're just sequencing DNA and RNA and then using the genetic code to say, well, it looks like it's going to be this is the protein it codes for.
Mm-hmm.
It's very hard to actually get to the protein and sequence it. It's a very difficult experimental process. So when they say predicted nuclear proteins, that means they haven't actually experimentally confirmed that they are nuclear.
Okay.
So there's guesswork there. However, how do you think they might predict whether a protein's nuclear or not? Just take a wild guess. I'll give you my guess. If
If
I
it
just
has,
gave you a DNA sequence and said, okay, this codes for a protein, tell me where it is in the cell. That's basically what they're saying.
I mean, they would probably be comparing it to known protein sequences, right?
Yes. And the word homolog. Are you familiar with the word homolog?
Yes.
Okay, it's a fancy word for similarity. The original meaning by Richard Owen is different than what the evolutionary biologists mean, but not it has some similarity. And so homology is basically similarity of form. So you can tell that just using the example of everyday things, uh, one airplane is homologous. It's similar to another airplane. You could tell that they have kind of the same structure and function, even though they're not exactly the same. And then you could say one car is similar to another car. And then you can even go down to models of cars.
Mm-hmm.
So, there's like the Honda Civic architecture, and then the Toyota Prius architecture, and then you have the Ford F-150 architecture. So you could say, a Ford F-150, there are all these cars that are homologous to each other if they're also Ford F-150s, but they might be different models of it. Does that sort of make sense?
Yeah.
Okay, so we are homologous. A lot of our body structure is very homologous to chimpanzees, gorillas, etc. We're
Mm-hmm.
not very homologous to plants,
No.
obviously. So, but we can, um, in the era of computation, we can actually see kind of homologous proteins, like a topoisomerase. Well, actually, you can kind of guess a topoisomerase in a human is homologous to a topoisomerase in an aspergillus.
Mm-hmm.
Um, the sequence, in terms of sequence, they're not identical, but they are relatively similar. I've actually looked at the topoisomerase. There's one listed here, Arabidopsis thaliana
Mm-hmm.
and Saccharomyces cerevisiae, and then human. And I've compared it using BLAST, and then another tool actually called Mega. And I might, if we have time, um, in one of our future meetings, I might show you Mega. But you could see that they're definitely in the same class. They're homologous to each other. So going back, how do we know that? How do we predict? Meaning we could be wrong, but we would say we have a reasonable chance of being right that it's a nuclear protein. So we look at some cell that we've totally blown apart and say, well, uh, what what what's in the nucleus of that? What proteins are there? So anything to do with transcription is probably in there, and replication. And so there's a whole suite of proteins involved in that. Um, can you name some of? Do you know off the top of your head some proteins involved in transcription and replication? Uh,
Yeah.
oh, go ahead, name them off.
Okay.
Just
Well,
it's a good practice for both of us because you'll stretch my memory too.
okay. Well, I know the DNA pol polymerase is
Very good.
really important. Um, and then I know there's a bunch of like proteins. I'm blanking on what they're called, but there's a bunch of proteins before the polymerase can like attach that kind
Mhm.
of prep the sequence and then
Um,
attract the pole. And then there's another one, the helicase. It's holding it open, right?
well done. Well done.
Yeah.
Helicase, polymerases.
And
Um,
obviously the
in the
topoisomerase.
top, hey, that's about the extent of my knowledge. I know there's something called a primase.
Yeah.
There also, not that I think that this is much in cellular biology or molecular biology, maybe in molecular. Some teachers will show that the helicase needs loaders.
Mm-hmm.
It needs to assemble the helicase right there. So well done. That's awesome. Um, yeah, that's a lot right there. Let's see what else. Um, I'm trying to think. Well, we can. Um, are you scheduled to study any molecular biology? Take any classes on molecular biology?
Um, no, not for, I mean, I have some more specific classes um for my senior year, not molecular, but I am taking developmental biology and cancer biology, which have to do with molecular to a degree. So,
Yeah. Well, molecular biology is a bit of a misnomer now
hmm
because it really does just deal with transcription and translation.
hmm
So you'll get very acquainted with all the proteins in the nucleus
hmm
in molecular biology. To me, the real molecular biology is biochemistry. So
hmm
molecular biology is a bit of a misnomer. Um, so um, so if they see homologs of topoisomerase in humans, we'll just say, hey, look, it's probably in the nucleus. So that's how I think they find some of this stuff.
hmm
Um, and at the time, they said they had 308 experimental. No, it's very hard to find. Oh, yeah, the database contains only 114 experimentally determined nuclear localization signals. That's really not a lot. Um, Considering the human alone might have 400 proteins, you would need at least 400, I mean, nuclear proteins, at least 400,
hmm
probably more. Um, and Homo sapiens is only one species. So you could see 114 experimentally determined nuclear localization signals is a very, very small sample of all that's out
hmm
there. Okay, so this is actually good to read. Um, you could unshare your screen.
Did you want to see the other website?
You can unshare your screen first
Okay.
before you transition to the other, just for. Um, in the meantime, I will hang on. Just let me know when you're ready to share your screen. Um, but I would like to see that other one, even if it
Okay.
gets that.
I can pull it up now.
Alrighty.
Can present.
So that gives us a feel for how much was known 11 years ago, and they've stopped tracking it.
Can you see this one?
Okay, let me bring it up. LogSigDB, a database of protein local. Yeah, look how many authors they have. One, two, three, four, five.
hmm
Okay, can you read the abstract for us?
Yeah. So Loxig database is a manually curated database of experimental protein localization signals for 8 distinct subcellular locations, primarily in eukaryotic cell with brief coverage of bacterial proteins. Proteins
Whoa,
must be.
whoa, 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 oh,
Oh,
whoa. I didn't know that. So it's not just nuclear localization. They have other. I didn't even realize bacteria had some sort of localization.
I guess.
I
Yeah.
just learned something. Wow. Sorry to interrupt you. Doing a great job, though. Go
Okay.
ahead.
Proteins must be localized at their appropriate subcellular compartment to perform their desired function. Mislocalization of proteins to unintended locations is a causative factor for many human diseases. Therefore, collection of known sorting signals will help support many important areas of biomedical research. By performing an extensive literature study, we compiled a collection of 533 experimentally determined localization signals, along with the proteins that harbor such signals. Each signal in the LOCSIG database is annotated with its localization source. PubMed references is linked to the proteins in UniProt database along with the organism information that contain the same amino acid pattern as the given signal. From LogSig database web server, users can download the whole database or browse search for the data using intuitive query interface. To date, LogSig database is the most comprehensive compendium, I don't know that word, of protein localization signals for 8 distinct subcellular locations.
Oh, great find, by the way, Katie. Can you
Mm-hmm.
scroll up? So they got up to 533, and that was
Okay.
2015, a year later. So you
Mm-hmm.
could see that, look, they had 5 co-authors to do this. So this is not trivial stuff. You can imagine how much work this would take. So
Mm-hmm.
you got a taste of it when you were just looking up aspergillus.
Mm-hmm.
So to be responsible, you would actually go. I could see why it would take that long. If they were doing this in a methodical, careful way, they would be reading all the publications, all 533 publications.
Yeah.
So
And
if
then
you
when
get
you try to click on the site, it says this. So it's unfortunate.
okay, so we can always try to contact the authors later, but this is good work. So you could take this down, and we'll talk a little bit where to go from here. Um, Let me get us back. Uh, so I compliment you on this work, and it is kind of it's a mixed bag. If you find out someone else is doing research that you've already done, you're like, well, I thought I'd be first, the first to find out. But it when someone else has already done research, at least there is interest in the field,
Hmm.
and you're not necessarily out in left field. Um, the hard part is trying to build on it. I mean, if you're really, really the first one to do something, you're going out on a limb because it could be risky. It could be a total dead end. On the other hand, if you wait till someone else starts working there, it's like, well, you weren't first to it, but at least um, it might actually bear fruit. It may not be a total dead end. So it's kind of a mixed bag. So um, so we're working kind of like detectives. We're not being spoon fed like we would be through a textbook where it's all laid out for us, kind of tidy. So I can tell you that maybe it's too hard to just do. By the way, when research is done, it's usually helpful if you could cite the work of other people that have already done work in that field and you build upon it. Um, so we were talking about gathering a collection of localization signals. Given that these websites are down, we might have. Would you be interested in just kind of rebuilding a portion of some of this?
Hmm.
Certainly not 533 or 308 or whatever numbers were thrown out.
Hmm.
Um, something that would be, say, 2 or 3 hours worth of work to get one localization signal from a creature.
Okay.
Because if you get to about five, that's already 15 hours of work.
Hmm.
Um, I mean, to find it, catalog it, and um, we've done aspergillus, and I have some ideas what to do. If we're only able to get a collection of 3 by the end of July, that's still fine. Um, since both of us are doing this kind of just by volunteering our time.
Hmm.
So let's see. All right, from the clues that I gathered on the first website, where do you think we might start? I'm just throwing this out there. I mean, we're acting like detectives now, and we're just kind of brainstorming. And I'll give you my take on it after you give any ideas that you have where we might go.
Um, I think maybe going to the Unipro website, like working with how we did last time, going to the Unipro website and getting a sequence for a certain species and then trying to figure out the localization signal through um, blast, like how we compared the two signals to find.
Okay,
Um,
so there are there are two there are kind of two dimensions of this. You
mhm,
could take the same protein across all species, like topoisomerase for all species that we can
mhm,
find, or you could take the same species and try to find all the proteins with localization signals experiments that we've had.
mhm,
And then if you really wanted to be just insanely comprehensive, which is not financially feasible for anyone on the planet right now, you take all the species on the planet and then all the nuclear proteins and catalog all the nuclear localization signals, and that probably fill out entire buildings
yeah.
of all the data. So given all the constraints we're working with, um, we might have to find out what's the easier path. Um, and I don't know the answer to this. Do we stick with one creature and try to find the localization signals for its nuclear proteins? Or do we kind of look at one protein like a topoisomerase and try to find localization signals across different species? Now, the problem is we know those signals exist, but have there been experiments that have identified them? And
Mhm,
and you could tell that that was probably like a year long, at least several months long experiment to find the localization signal in aspergillus. So um, let's just try to find an easy route, and maybe there's no easy route to that question. And we might just have to do what little we can. I mean, it's not going to be a total failure if we just say, hey, look, this is all that we could gather. It's just kind of it's fragmented information. It's like we know that there's a jigsaw puzzle that can be assembled, and we only get 2 out of 100 pieces,
mhm,
and that might be all we could do, and that still counts for something because if we publish research on this, it'll tell the rest of the world, hey, we've we've looked at what's been done, and so don't necessarily try to duplicate what's already been done. There are areas that there's so many wide open areas that you could do experiments to uncover this. So I don't have the answer to that. So in the first website, it listed a whole bunch of creatures. I already can guess which one you might be interested in.
mhm.
That'd be the fruit fly. By the
Yeah.
way, did you physically have to take apart those things in the lab?
Yep.
Oh,
And it was, well, it wasn't, it's not as bad as you think. It's larval dissections. So you just start dealing with a little yellow blob.
oh my goodness! But it's so tiny. I
It
thought
is really
this
hard.
was
We have to look at it under a microscope while we're doing a dissection.
well, well done, well done. Um, That's why I'm not a field biologist. I couldn't deal with creatures. um, okay, so would you like to try to find a localization signal for um, Drosophila? And I
Yeah.
don't know where to begin. Do
Yeah.
you want Okay, so we have half an hour left. Uh, why don't we give it a Why don't we give it a try?
Okay.
Okay, so I'll go first, and I'll bring up. Okay, I may totally botch this, but uh, hopefully we'll all learn something from the process. So now there is a button on your StreamYard screen where you
Mm-hmm.
can get it to expand so you can actually see. Um, and let me before I tell you how to do that. Okay, so okay, so there's like this little. Let me see if I could show it to you. You're going to get a whole bunch of screens here. Uh, I don't know if you could see my pointer.
Yeah.
Do you see it's going into that little square?
Yeah.
When you click that, you can expand. You can get the focus. You'll get a little bit better. You can see things a little better. And all you have to do is press escape to go back to your normal screen.
Okay.
So hopefully you could see things a little clearer. So now let's go to
Mhm.
Unibroad. Okay. So since we've done topoisomerase, we'll do top two. Now, top 2A is specific to mammals like humans, etc. So I'm just getting a general topoisomerase. Look at that! Did it read our mind? Look at the first entry. What do you see what I see?
Oh wow. Yeah. I think it was listening to you.
That's kind of creepy.
Mm-hmm.
Okay, so all right, I'm just totally guessing here. Establishment of localization. Uh, let's say no publications. Yeah, it's giving me no publications. All right, so don't lose heart. Even for the human topoisomerase, there were papers published on it, but it just wasn't in Uniprot. Sometimes it's not linked there. So
Mm-hmm.
what can we resort to?
A
Well,
Google search.
yeah. Um, okay, so we might not. This AI overview might not be the most accurate. Um, so they have one for topoisomerase one.
Mm-hmm.
Okay, so I think topoisomerase one. It's different, obviously, than topoisomerase two. I think topoisomerase one is it just cuts one strand of DNA. Topoisomerase class two cuts both strands.
Mm-hmm.
Um, wait, pause. You have topoisomerase 1, 2, 3, 4, 5, and 6, but they fall into two classes, type 1 and type 2.
Mm-hmm.
It just so happens, I think topoisomerase 1 falls in is a type 1, and topoisomerase 2 is a type 2. Where it gets funny is what happens to 2, 3, 4, 5, and 6.
Mm-hmm.
All right, so I'm pretty sure. Okay, so they have one with topoisomerase 2, and I'm just going to see. Hmm, wow. they're actually looking inside. I think these. Well, have you done any immunofluorescence?
I have, yes.
You have. I'm
Mm-hmm.
jealous. So this isn't an. You could teach me what this means then.
I maybe, yeah.
All right. So, oh my goodness, look at how hard they had to work to just figure this out. This is
Mm-hmm.
this is how hard they had to work to find out what the nuclear localization signal is. Oh my goodness, hats off to them. All right, so that was topoisomerase one. I should have said, see if they have a type two. Oh, maybe we hit the jackpot here.
Oh.
Oh, and we got a good one, Asharov.
Mm-hmm.
Do you remember do you remember the name Joe DeWeese?
Yeah.
Asharov is his colleague.
Okay.
So this is going to be, oh wow, in situ localization DNA, a major. Um, can you see this clear enough to read it for us?
Yeah, I can read it.
Can you hang on just 1 second?
Okay.
I hope you won't be offended, but I want to get a dessert while you read this. I'll
Okay.
be right back.
I guess I'll read it. DNA topoisomerase 2 has been immunochemically identified on protein blots as a major polypeptide component of the Drosophila nuclear matrix, pore complex lamina fraction. Indirect immunofluorescence analysis of larval cryosections have confirmed the nuclear localization of topoisomerase 2 in situ. Are you back?
Yes, I went to the fridge. I'm going to tell you what I got. Okay, maybe
Okay.
just for the sake of the audience. All right, people have known I've been on a special diet, um, a low sugar diet, but I've been cheating on my diet, partly because I got so thin. I said I actually have to start eating carbs. I was on um, a carnivore diet for a while, and I lost so much weight. I really was trying to fight pre-diabetes,
Mm-hmm.
but then the church gave me all these. They gave me, look at all the
Oh,
donuts,
wow.
and I have a banana. Okay, just to tell you what I just had a craving for it. So um, I actually look way healthier now than I did. But if I if I didn't go off the carnivore keto paleo diet, I was going to turn into a skeleton. So that's a discussion for a totally different day. But just so you know what I was up to. So let's go. Uh, um,
Okay, I think I misheard you when you were. I thought you wanted me to read it while you were gone, but I'll read it again.
okay, all right. So let me get our screen share going again.
Okay.
Oh, that's all right. We're having. Are you having fun?
I am, yeah.
Okay, all right. If we're having fun, that's all that counts for today. All right, go ahead. Thank you.
Okay. DNA topoisomerase 2 has been immunochemically identified on protein blots as a major polypeptide component of the Drosophila nuclear matrix pore complex lamina fraction. Indirect immunofluorescence and analysis of larval cryosections have confirmed the nuclear localization of topoisomerase 2 in situ. Although apparently excluded from the nucleolus, the topoisomerase protein is otherwise distributed throughout the interior of the interphase nuclei. Similar immunocytochemical studies performed with permeabilized whole giant cells from third instar larval salivary glands have shown topoisomerase 2 to be largely restricted to the polytene chromosomes, Upon nuclear disassembly during mitosis, the topoisomerase polypeptide appears to redistribute diffusely through the cell. Faint immunofluorescence staining of mitotic chromosomes is also observed.
Well done. Do you feel you can understand a lot of that in light of the last few weeks that we've studied together, plus your background in working with Drosophila? I mean, I
um,
can't say I understood everything. We can go through it. This is actually, by the way, have you heard the term journal club?
yes.
So we're doing a basic journal club here, and we're getting more
Mhm.
familiar with the field. And you can obviously see that there is just too much to learn. You know, you can only specialize in one little section of biology. So do you want to
Mhm.
go through this carefully? I
Sure.
would.
Yeah.
And you could this time you can actually inform me a few things. Okay. So we could just start from the uh, the beginning of the abstract again. I'm sorry to
Okay.
make you read it over, but this time I'll interrupt you.
Okay. DNA topoisomerase 2 has been immunochemically identified on
Okay.
protein blots. Oh,
Okay. Let's stop. Have you
okay.
done any immunochemical identification? I've never done that. I know
Yeah.
that there's something. Why don't you explain it to me then?
Okay. Um, well, I think it's immunohistochemistry or something like that, IHC. Um, and you're staining whatever you want to look at, like the tissue that you want to look at. So in the experiments that I would do, I was looking at um, larval eye discs, or it's the precursor to their eye. And so you stain it through a process of staining using two different antibodies, I believe, so that you can look at it under a fluorescent microscope. And so they're probably using like GFP. And most of the time, it's to see if certain cellular processes are going on. So, like, you can see if mitosis is occurring through this kind of staining.
Hmm. Awesome. I mean, I've heard about immunofluorescence and and I heard it has something to do with antibiotics. That's the extent of my experimental knowledge. So you're ahead of me on that. Have you done work on protein blots?
Like a Western blot? Is
I
that the
guess
same
that.
thing?
Yeah, that's what I've heard. Um, and that always cracked me up when they would say northern, southern, western, eastern
Mm-hmm.
blots. I think the first guy was his real name was Southern or something,
Oh,
and then they just started making fun of it
that's funny.
because they called it a Southern blot, and they started coming up with Western, Eastern, and Northern.
Hmm.
Okay, so um, go ahead and keep reading. I'm sorry to interrupt you.
It's okay. Um, as a major polypeptide component of the Drosophila nuclear matrix pore complex lamina fraction.
Okay, so you know what a polypeptide is? It's it's like it's a proteins are polypeptides, but not all polypeptides are necessarily proteins. It's
Hmm.
basically an amino acid sequence joined by the alpha peptide bond, which we'll study in organic chemistry
Okay.
and biochemistry. Component of the Drosophila. Okay, so nuclear matrix pore complex lamina fraction. So I understand nuclear,
Hmm.
and there's a matrix pore complex. A pore complex is at the membrane
Hmm.
of the nucleus, I think. And I don't know why they call it a lamina fraction. So I don't understand that term. If you understand the term, just tell us. Okay, indirect immuno, yeah, indirect immunofluorescence. So you have done immunofluorescence, right? That's GFP.
Hmm.
GFP is FP. GF is P is for fluorescence. No, F is for GF. Oh,
GFP.
F is fluorescent. P is for protein. I don't know what the G is.
Probably green.
Oh, okay. Thank you. So you've done immunofluorescent analysis. Okay, a larval cryosection. So you've worked with a Drosophila larva, right?
Yes.
Does the word cryosection mean anything?
I haven't heard that before, but I can make a guess that it's maybe it might be like the different precursors for each um developing part of the Drosophila, because you have like the eye discs, you have the wing discs, and I think somewhere else they like, I don't know if it was in this one or the other one, but they talked about what part of the Drosophila that they stained.
Well, see, this is
The
also good.
salivary glands, sorry.
Yes. Well, this is actually interesting because do you have access to the researcher still um, that you work with when you're doing Drosophila?
Oh, yeah.
So you could ask them what a cryosection is.
Okay.
I mean, you don't have to do it for this, right now or anything. I'm just saying, hypothetically, you have. This is the good thing about working in a laboratory in your specialty. You have access to people that can explain all the terms, you know.
Mm-hmm.
Um, and so that's how come we end up being in certain specializations. It's just easier because then you can read the papers and you have someone you fall back on who can look stuff up for you, or they could just tell you. Okay, go ahead.
Okay. Where was I? Indirect immunofluorescence?
Cryosection.
Oh, the cryosection. Okay. Um, have confirmed the nuclear localization of topoisomerase 2 in situ. Although apparently excluded from the nucleolus, the
Are you
topo
familiar with the nucleolus?
um I know it's a part of the nucleus.
Okay, that's good enough. Let's go on.
Okay. The topoisomerase protein is otherwise distributed throughout the interior of the interphase nuclei.
By the way, I learned something.
Mhm.
I didn't realize the nuclei changes depending on the phase of the cell.
Yeah.
Wow. I'm ashamed because I did take 2 semesters of cellular biology. I should know that. Oh well.
Similar immunocytochemical studies performed with permeabilized whole giant cells from third instar larval salivary glands have shown topoisomerase 2 to be largely restricted to the polytene chromosomes.
Okay, stop. So you said, did you work with salivary glands?
No, I didn't even know that you could get salivary glands from a third instar larva. That
So what is
surprises
that third instar larva?
It's the um stage in larval development right before they turn into a fly, I guess you could say. We know that they're that way because they're usually moving up the sides of a test tube.
Hmm.
Whatever they're encased in, they're like trying to get to the top of it. They're or no, other way around. They're moving down to get into the food.
Hmm. See, this is great because now you're helping teach me what this means,
Mm-hmm.
and so this was actually kind of um, interesting that, like what you said, you didn't even know that they could grab cells from the salivary glands.
Yeah.
Wow. Okay, go ahead. So salivary glands
have shown topoisomerase 2 to be largely restricted to the polytene chromosomes.
I've never heard that term before, a polytene chromosome.
I haven't heard it either.
Okay.
Um,
Okay, go ahead.
okay. Upon nuclear disassembly during mitosis, the topoisomerase polypeptide appears to redistribute diffusely throughout the cell. Faint immunofluorescent staining of mitotic chromosomes is also observed.
Okay, so let me see if it is a PMC. Do you see my cursor here?
Yeah.
All right. If you see PMC, a lot of times you can get the entire publication for free
Mm-hmm.
and see if we can get it. Ah, and see if we can look. And oh my goodness. Oh, they photographed this. Oh my goodness.
Mm-hmm.
um, this isn't just like a regular text. You're gonna have to. Okay, I'm not gonna say you have to do this, but since this is a creature you're already familiar with, and you're familiar with topoisomerase too, do you think this would be? Um, I would be curious. Okay, so what I want to do is get the sequence. So can we put that as a project to find out what the actual sequence is?
Yeah, you can send me this article. I can look at it and
Okay.
read through it.
Yep, I will do it. So I'm going to stop the screen share. Maybe we'll stop early for a change. Um, so you could work on the. Um, how about we meet backstage shortly?
Okay.
We'll close this recording down. Then we could talk the specifics. What the next steps are. Um, so this is a very beneficial session for me. I didn't I didn't know a lot of this stuff, and you have lab experience. I'm jealous.
I do. It's fun working with fruit flies.
Um, I still can't get over that you could work with them. They're so tiny. It's just wow. The techniques you'd have. Oh my goodness! When I was at the NIH taking a class. Um, the professor invited me to her lab, and we were walking around. She's going to show me her lab station, and there are all these fruit flies buzzing around the lab. She said, "Yeah, uh, that other section they do research on fruit flies, and some of them escape.
Mm-hmm. Yeah, that happens.
They're just buzzing around the laboratory. It's like wow. Um, so I respect the field biologists and the guys that do the experiments. Uh, I just kind of do the abstract stuff and do the bioinformatics. I could just sit in my chair and kind of do stuff. Um, so well done on that. So um, there were. Why don't we look at? Okay, so we had Homo sapiens. We have the topoisomerase 2 on that. You also have the topoisomerase 1 on the fruit fly. I guess it wouldn't hurt to find out. I'd be very, very, very curious if the localization signals for topoisomerase 1 and topoisomerase 2 are the same or different.
Mm-hmm.
I will tell you, an evolutionary biologist said topoisomerase 1 is so different from topoisomerase 2, sequence
Okay.
1. I would be just curious. So um, so we were talking about whether we go, I'll say, do we look at one kind of protein across species, or do you look at one species and look at all the possible proteins?
Mm-hmm.
I would be curious how many kinds of proteins they've characterized in Drosophila that have nuclear localizations. So, that's going to be kind of hard. Short of actually naming every protein you could think of, and then in the list of all proteins in Drosophila, and Googling it, I don't know how to go about finding, localization signals that have been experimentally found. That'd be a very challenging task. So that would be something just to do when you don't have anything else to do, I guess. So I don't have much. This has been very productive. let's just pause a little bit. Do you like? Would you like to explore this paper more?
Yeah, definitely send it to me. I'm gonna read it and look at what they found. Usually, from my experience, the especially the people who are writing about Drosophila are very hard to understand, but um, I can figure it out. Don't worry.
Okay. Um, so, what is our main goal for this particular paper, and maybe the topoisomerase one?
To figure out if they have a nuclear localization signal.
And
Um,
what the sequence is. I want to
yeah,
find
what
that
this
sequence
was.
because then
Yeah.
we're going to compare it. We can compare it to human
Mm-hmm.
to an aspergillus.
Mm-hmm.
I have to work hard to get the one record of the human nuclear localization signal. It's in a printed copy that's not accessible through the library I have access to, which is George Mason. I'm an alumni of George Mason, and I
Mm-hmm.
can't believe they don't have it in their collections. I have to physically go to the library because it's an old printed article,
Oh wow.
26 years ago, and I yeah, I'm not looking forward to that. Otherwise, if it were available electronically, I'd just go ahead and give it to you, but I can't. Now, I did show you where I had a nuclear localization for topoisomerase, but now I'm beginning to wonder if that was just uh, by inference. I don't know that I necessarily found it in an electronic copy. I need to go verify that I actually have the right sequence. Um, Now, I might send you this paper now that we're talking more about this on the difference between a bipartite and I think it is monopartite. I knew bipartite is a word. I don't know if monopartite is a word. So there are at least two classes of nuclear localization signals. One is where you just have a very simple individual sequence that's
Mm-hmm.
made of basic amino acids. Do you remember what they are? Lysine, arginine, and
Histidine.
histidine. Um, you might have a just like when you saw that video on the nuclear import complex.
Mm-hmm.
It might be short. That'd be monopartite. But sometimes they'll have two of those spread apart by a certain number of amino acids, and so you have basically two monopartites kind of stacked close together in proximity with some intervening
Mm-hmm.
sequences in between. That's a bipartite. And I saw a paper on bipartite localization. It might be helpful just to also read it, but I wouldn't make that a high priority. So when I send you the email, I'll just suggest what I think would be fruitful for your, for the project that we're trying to work on. Um, but you're an equal partner on this adventure, so this isn't like an instruction. It's just what I think might be fruitful. That's all it is. So
Mm-hmm.
um, we'll do. Okay, so would you like to summarize anything you found interesting in this discussion or so far today?
Um, let's see. What did we talk about? Well, I just thought the technique that they were using for the Drosophila, like, well, I don't fully understand it to be quite honest how, but I guess I'll read about it. But um, using a technique that I know about, immunohistochemistry, to figure out this localization sequence is pretty cool to me. But summarizing everything, I guess, um, um, like of what we did today, right?
Well, I mean, just free thought. I mean, I'm not trying to
So,
force. It's kind of good to for me to see how you know what takeaways you got from this adventure today.
um, there are some previous research. There's
Yeah.
some previous research done in localization signals. but it doesn't seem like there's a lot. So it'd be interesting to see if we can see if those websites, well, I guess they're not really available, but it'd be interesting to know what those scientists are doing now, honestly.
Yeah.
Yeah.
Well, I'll give you my take on it. It's mostly which you. It's also your take. This actually was very eye-opening for me to see how little we know
Hmm.
for such an important process. We've actually had so few experiments to actually, actually find the nuclear localization signals. And when we were scrolling through that one paper that had all those, um, um, uh, all of those fluorescent figures, I was
Hmm.
like, this is really hard research. I mean, I'm not faulting the scientists or the community for having so little data. It's just because this is really hard. I mean, the agony they have to go through to be able to identify uh, where the localization signal is in just one protein in one species. Um, wow. I mean, hats off to them. That's the thing that, so it did strike me how little we've actually found on such an important topic, but then also the magnitude of difficulty to even get just what little we have. I mean, this is just amazing. Um, I can now understand why, like, um, uh, Dr. Osaroff, who's Dr. DeWeese's colleague, um, and I've been privileged to be Dr. DeWeese's colleague too, kind of, but a very junior one. Um, I could see now how these guys could be spending their entire lifetime studying one protein class, like topoisomerase 2, whole
Hmm.
lifetime. And then they have I just FYI, um, Joe attends like there's like a monthly or weekly conference on topoisomerase. That's all they talk about. 1,000 researchers around the world talk about topoisomerase. Then they meet again. This is how much energy is invested on just one protein. Just trying to understand it. It's amazing. Um, so those are my thoughts, and I was blessed today to see you be able to walk me through some parts about Drosophila, which I didn't know about, and also the lab techniques. So I feel privileged to know a fellow researcher here.
Yeah.
So are there any closing thoughts? Otherwise, I'll just close the recording down.
Yeah, we can just close it now.
All right. I'd like to thank all of you who joined us on this adventure of science. Um, and till Lord willing, till we meet again, take care and God bless you all.